Hello, everyone, and welcome to today's webinar, Acceleration of Neuroscience Research Discovery by Incorporation of Large Area Volume Microscopic Data. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions that we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Anastas Popratiloff, Lead Research Scientist, George Washington Nanofabrication and Imaging Center, and Professor of Anatomy and Cell Biology, George Washington School of Medicine and Health Sciences. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Anastas, you may now begin your presentation.
Thank you, Christy, for the introduction. I want to also thank Leica Microsystems for inviting me to give this presentation today. I'm going to try over the next hour or so, I'm going to try to make a case and to convince you that neuroscience research discovery can be accelerated by incorporation of large area and volume microscopic data. This way, I'm going to go to my first slide. We live here. I want to emphasize that we live in really interesting times where the technology impacts our lives and habits in very rapid pace. Digital imaging, for example, has become a very integral part of our existence and invited all aspects of our life. Google Earth, as here, for example, shown, it's a really prime example of multi-layered image integration, and this tool has provided us as consumers to zoomable layer data, which enables us the orientation and navigation in space. So a characteristic feature of such data approach is that the pixels are linked to a global positioning map that's known in most of the cases as GPS. Another feature is that the final image is much larger than the single field of view of the device generating the image data. This example from our north part of the GW, George Washington University Foggy Bottom campus, also shows the layers of data such that the important features are placed in the image map in 3 dimensions. This itself was an example of layered data where the layers are precisely linked to the global positioning map, but also to each other. But all these layers derive from relatively independent data sets. In the scale of human lifespan, the data represents flow dynamics, so that we can assume that pixels represent Google Earth are static. In addition to that, layers of dynamic data can be also linked to these layers, such as clouds, radar, winds, etc. Altogether, this imaging approach changed how we live, travel, even what to wear next day. The reason behind the success of this approach is that our brains can manage relatively easy, temporarily integrate the zoom out, which represents the context, and the zoom in, which represents the resolution. So now, nowadays, most of the microscopes are capable of generating layered data sets. This is an example of our Leica SP8 confocal microscope that we have in our facility and can generate layers of data, providing information about spectral characteristics, fluorescence lifetime, 3-dimensional volumes. This altogether is enabled by software known module known as Stage Navigator. A key element of this kind of instrument is that they are equipped with a scanning stage that can fit space information into the data set, making it possible to use the image data also as a navigation tool. Another key element of these instruments is that by capturing and stitching together adjacent images, they can generate two-dimensional and three-dimensional data sets beyond and above a single field of view. Taken together, such instruments would ideally allow scientists to generate something very similar as Google Earth, where the image data serves as a navigation tool, while layers of image data represent different elements of the object under study. Moreover, assuming a low dynamics of the sample for the life of the experiment, one can also link dynamic data into layer of static data. Interestingly, whereas Google Earth changed individual and social behaviors, the ability of the modern microscope to generate similar data sets did not drive much demand for implementation. To my experience, most of the limitations are data management driven, but there is still relatively little understanding of the actual value of data sets that exceed single field of view, and more importantly, the behavior and appreciation of how to structure complex data sets. In this slide, I'm trying to outline several important, to my opinion, elements in building complex microscopic sets. So first of all, the low-resolution, high-speed data collection is necessary to generate object positioning map. This is the first layer of data that serves as a navigation tool. The resolution and speed are inversely corrected, as many of you know. But the question here, the fundamental question is, what is the minimum resolution necessary to achieve reliable navigation through the sample? Next, the acquisition of higher spatial resolution layer aligned with the low resolution positioning map. Frequently, the axis, the alignment, and other elements of the system changes with the magnification by changing magnification, resulting in the data XY shift, which will require alignment of the layers. Then, building a layer data set by fusing low and high resolution data set. High resolution data is always more expensive to generate, store, and structure. Layer data allows for capturing the low profile real estate at low resolution, while the only high profile areas are acquired at high resolution, but still linked to the low resolution map. Use of image data as a navigation tool, or with other words, interaction with the scanning stage via positioning map image. This is important to receive a feedback from a low resolution image, which is critical skill enabling the investigator to navigate quickly within the sample with reliability. Then after we have new things that are challenged for us, like dealing with tiling and stitching artifacts and shifts. There is a whole new dimension of image artifacts deriving from stage movements or as additive function of otherwise minimal aberration and image distortion. And everybody has to be aware of that and to prevent as much as possible. Then fusion of very large image data sets at dramatically different resolution. And finally, understanding really the signal generation of the detector and post-processing downstream to the pixel grayscale value. So with this image from Allen Mouse Brain Atlas, I try to emphasize on the importance for the solid neuroscience research to sample from functionally precise group of neurons. Here, for example, the neurons in layer 2-3 of the primary motor cortex are functionally different than the layer 2-3 neurons of the adjacent primary sensory cortex. The cytoarchitecture map, which is on the left side with the initial staining, includes cell density, is what a neuroanatomist needs as a basic information to define border of the cortex. On the other hand, most of those neurons will send axons beyond the cell body and dendrites and much beyond the field of view of single field of view, which is taken at desired resolution for, let's say, for example, dendritic resolution. In other words, the ideal data set will include volumetric data of the entire brain and all elements of the neuron. Unfortunately, the brain is not opaque, and the technique that allows for imaging the entire brain are resolution limiting. So, to surmount the transparency problem of the brain, neuroscience research experienced the boom of the so-called clearing techniques. The common of those techniques is that they can bring the optical properties of the brain to predictable and uniform transparency with non-refractive index. The non-refractive index allows for selection of proper optics and thus reduction of spherical aberrations when large volumes were captured with confocal microscope. Tissue clearing with benzyl benzoate has been introduced to the anatomical research by the German anatomist Spaltenholz, who realized that organic solvents can remove the lipids and thus whole organs can be cleared and structure studied. So despite the number of techniques available currently for clearing the brains, we use the recent modification. We have been refining imaging for about 5 years now, as we started with 10x magnification, 10x objective with 0.45 numerical aperture lens. In this example, the entire embryo has been cleared with BABB, immunolabeled with marker for neuronal-specific filament UJ1. The data represents a volume of the entire embryo that is about 1 millimeter thick. The final volume is produced by stitching 3 by 3 adjacent volumes, which as a final result give a resolution of a cranial nerve, while also provides us with the morphology of the embryo. In this case, the gray channel represents autofluorescence, which has been spectrally separated from the TUJ1 fluorescence by linear spectral mixing. In this case also, one has to really think of what is available as imaging technology. For example, 0.45 numerical aperture can provide with the desired working distance to image a volume of a millimeter, but at the same time will compromise the resolution necessary to resolve a single axon. We've been further exploring options of how to overcome this limitation by testing different objective lenses. We identified several multi-immersion lenses that would allow us to improve the resolution with numerical aperture ranging from 0.75 to 0.8. Moreover, these lenses may be corrected for OL, refractive index, which perfectly matches 1 to 2 BABB refractive index. In this example, 1/2 of 10.5 mouse embryo has been imaged with 25x 0.8 all immersion lens using a spinning disc technology. The working distance is only about 500 microns from the cover slip, which requires imaging only 1/2 of the embryo at a time. However, we gained the dramatic improvement in the X and Y resolution, allowing us to analyze single axon. Despite that, the perfect match of the refractive index, this type of data is compromised in the Z-axis, since the spinning disc technology does not perform well away from the cover slip. Also, a methodological driving force for us was the question of how individual neurons and their axons develop in mouse model of a human disease. Our group, led by Antonio Lamantia, was generously funded by NIH program grant to utilize mouse model of a human developmental disease clinically manifested as DiGeorge syndrome, or also known as 22q11.2 deletion syndrome. I will not go into a great detail about the disease and the mouse model because this talk is mainly focused on the methodological aspects of the work, but would like to highlight several important points that drive our experimental design. There is remarkable similarity between symptoms presented in the large deletion mouse and the 22q11 deletion syndrome in humans. However, our main focus for this work was dysphagia, which we believe involves compromised development or maintenance of the cranial nerves. So, whereas the human disease is caused by spontaneous deletion of a 3 megabase pair region in the chromosome 22 in the region specified as Q11.2, the more frequent deletion includes a reduced 1.5 megabase pair region. The mouse ortholog of this region is situated in chromosome 16, as the mouse model that we utilize is popular as a large DEL and corresponds very well to the typical human deletion. So the work of Beth Karpinski using TUJ1 labeling from the whole embryo identified that development of the cranial nerves is compromised in the large deletion. However, we wanted to move these studies to the next level and to investigate the contribution of single motor or sensory neurons, their axonal trajectories to the branchial arches, and the central processes entering the neural tube. In order to address this kind of question, we turned to neuronal tracers. We turned into an approach that I had been previously developed to study the distribution of gravity-sensing organs, nerves, using chicken model. This slide is an example of Alexa-conjugated biocytin simultaneously injected in sacral and utricular nerves of 5-day-old chickens. The injections resulted in complete labeling of the central process of these sensory neurons within 1 hour after the application. However, this slide also illustrates how the data is presented in an era of limited technology, analyzing individual serial sections with a hope to put an accurate three-dimensional model of the fiber distribution. This slide here represents a high-power example of the quality biocytin tracers can provide in resolving synaptic terminals and axons. In this case, high-power images were taken from vestibular nuclei receiving otolith terminals, allowing identification of synaptic terminals and the primary vestibular afferents. However, despite we kept track of the location of these high-power images, the relation to the overall context is missing. Obviously, to interrogate the abnormal pathfinding in single neurons from embryo, the previous approach is not an option. Therefore, we combined clearing and tracing with the aim to generate high-resolution volume from the region of the embryo that includes the peripheral target, with, in other words, the mandibular branchial arches, the trigeminal ganglion, and the corresponding regions of the neural tube. So, briefly, the embryos are removed from the uterus and kept alive in medium. The tracer is injected in the branchial arch under visual control, and the embryos are allowed to survive in the medium for an hour before being fixed. No other visualization step is necessary. The challenge for imaging this large volume is that the region of the tracer, which is within the white box in this particular image, is much brighter than the region containing retrograde label neurons within the red box, for example. The conventional way of photon imaging using multi-alkali photomultipliers and continuous signal processing followed by digitalization, in principle, results in either saturation of the injection site or a loss of the data where weak labeling is present. In order to surmount this problem, we used Leica hybrid film-gated detectors in photon counting regime and excited the sample with 80 MHz picosecond pulse source. Using this approach, the pixel brightness represents the number of photons arriving at the detector within a given period of time. To maintain the linearity of data, we lower the fluorescence output by decreasing the output of the excitation source, and we use temporal integration to achieve reasonable photon counting. The overall data and outcomes after applying this approach has been presented previously in previous LabRoots webinar by our program postdoc Zara Motahari, and you can follow the link to this presentation to view the webinar. However, the most important is that by proper aberration correction, matching the refractive index of the ABB with like a 20x 0.75 oil immersion lens, you can produce image quality, allowing for further image processing and analysis without compromise. The dynamic range is excellent, allowing for visualization without saturation of the injection site and the finest process of the trigeminal, sensory, and the motor neurons. I want you to consider that this volume is also produced by close to Nyquist pixel size in this spacing. As a result of smooth image data, the next step of structuring the image data, as in this case tracing the process and segmenting the cell body, is greatly facilitated without obstructions. The final outcome of this study is that the axonal outgrowth is compromised in the large deletion. The upper panel represents trigeminal sensory neurons that have been traced and for analysis, whereas the graph illustrates the higher frequency of branching and looping in the large deletion animal. However, in order to move this type of studies to the next level and to investigate the axonal outgrowth in detail, we need much higher resolution approaches. A well-known limitation of the light microscopy is the limited resolution, limiting around 150 nanometers in the best case scenario. At the same time, electron microscopy offers much higher resolution, but the trade-in is inconvenience, tedious sample processing, and until recently, very limited field of view. We've been developing light microscopy approaches that can generate image sets beyond single field of view, as you can see on the upper left panel here, representing a large area of a cover slip containing trigeminal sensory neurons at very high resolution. The registration is achieved with the Leica Navigator module, which allows not only to capture a large area, but also to integrate low and high resolution imaging data and to use the low resolution as navigation tool. The integration of Leica's file format to a very similar software that is used in our Thermo Fisher FEI electron microscopes allows us to easily integrate and align light microscopy and electron microscopy data as shown on the upper right panel. We also work on different approaches, so as the images on the bottom left, for example, illustrate sample that has been imaged previously with the Leica Navigator, and you can see the high power magnifications in the lower right panel, increasing magnification. In this case, we use photoresist printed patterns on silicon wafer, which silicon wafer has been used for substrate to grow the neurons on. So now, this movie illustrates the resolution and the field of view that can be routinely achieved from cell culture experiments using Leica Navigator module. These are explanted trigeminal ganglion cells, in this case from the large deletion animal, where labeling with neuron-specific filament UJ1 shown in red and actin shown in green. I would like to underline here that the necessity of generating high-resolution large area data is that in order to quantify the axonal growth abnormalities, one needs really the entire axon from the cell body to the growth or growth cones. So the next movie shows the image set presented on the previous slide, but the data has been used to generate high-resolution scanning electron microscopy image in a similar way what has been used to generate the light microscopy data. As you can clearly see, the topology, EM, which is achieved with Everhart-Thorney secondary electron detectors, add an extra dimension to the confidence in analysis that includes membrane morphology and eliminates ambiguity in discriminating overlapping from branching axons. Now, I would like to move from the trigeminal system to the hypoglossal nucleus, which is a brainstem motor nucleus containing the neurons innervating the tongue. Again, our interest in this target are because of the feeding abnormalities clinically documented in the 22q11.2 deletion syndrome. Our overarching hypothesis for this study was that the neuronal survival is compromised in large deletion, thus the quality and quantity of the tongue innervation. Tom Minor developed this very nice breeding approach that included crossing of the large deletion mouse with the cholinestyltransferase link GFP reporter line. Thus, all motor neurons in the brainstem are glowing GFP. This approach allows us to cut 100-micron thick sections and to capture at high resolution a volume of the entire hypoglossal nucleus, which is present at the center of the images. The box in the lower left corner of the images to the left represents a single field of view composing the final image sets. The high resolution gave us the ability to apply threshold-based and seed point segmentation image analysis approaches and to extract the cell volumes and to use the cell volume as a biomarker for neuronal degeneration. After pulling together hundreds of neurons, we found increased variability in the cell size in addition to reduced mean volume. Despite the fact that this data passed the significance, this data was more indicative rather than determinative of neuronal degeneration. We further used single biocytin injected cells, as it's shown on this low power magnification, to analyze the changes in the dendritic branching and the morphology as a biomarker for neuronal degeneration. So here is the lower magnification map of the entire brainstem section, and you can see three neurons are labeled, two on the left side, one on the right side. First of all, this map indicates that these are motor neurons. Second, we can validate that these are hypoglossal neurons by following the axon to the intramedular part of the hypoglossal nerve, which is going from the cell body to the lateral and ventral of the section. This allows us to produce very high quality. These are 300 micron sections, so most of the dendrites are within the section, and it can give us, a very nice picture of the normal hypoglossal motor neurons, quite variable from neuron to neuron, but we can do the segmentation of the dendrites, and eventually we can use this as a matrix for diagnostics. This is a counterpart of hypoglossal motor neuron taken from the large deletion animal. Low magnification, this is usually the first step. This is the low magnification map indicating the neuron and clearly axon going into the hypoglossal nerve. Very important here is to notice if you move a little bit away of the hypoglossal nucleus, you capture completely different quality of neurons. So this is the first thing that we need to verify, that all the neurons that are patch clamped here, they belong to the hypoglossal nucleus. So now this movie, produced by stitching 9 confocal volumes from this 300 micron brain slice, represents a single biocytin injected hypoglossal neurons. When we look at the high resolution, the dendritic trees clear morphological changes of neuronal degeneration, such as these corkscrew dendrites that can be observed. Although not all dendrites show signs of abnormality here. In general, to produce such an outcome, the images need to support single dendrite resolution of less than a micron. In addition, the high resolution is necessary to support the next step of the structuring of the image data, as in this case, filament segmentation and determination of the dendritic branching pattern. So although we've done all the right things, the qualitative data is more dominant than the quantitative data. The quantitative analysis of complexity, length, straightness did not reach significance. But a closer look, however, as shown on the lower graphs, offers some indicators suggesting a compromised complexity of dendritic tree. But this is happening at the very distal dendrites. At the same time, the qualitative changes in the more proximal dendrites cannot drive the data to significant differences. Thus, the light microscopy is suggested as a possibility of neuronal degeneration, but at the postnatal day 7, this may be just unfolding pathology. We decided to take a different approach and to use high-resolution electron microscopy imaging to define the abnormality in the hypoglossal nucleus, neurons in the large deletion, and to test directly the hypothesis that there is a sparse neuronal degeneration occurring in these neurons. The conventional TM, however, will represent a sampling challenge since we are most likely dealing with onset of pathological process where a spike of rare events will indicate pathology. So we need to increase the sample area and the sample size. Moreover, if the system is compromised by neuronal network load-dependent neuronal degeneration, the abnormality will be more pronounced at the later stages of the postnatal development. With the field emission electron sources, the implementation of solid-state detectors, the scanning electron microscopy now offers new dimensions for imaging at high resolution. This example of paper that influenced me a lot, that utilizes serial sections that are placed on silicon for SEM imaging, as the authors modified little the classical staining procedure to enhance the electron backscattering from the sample. In principle, the SEM will produce the same quality but inverted intensity relative to the TM. The use of SEM as alternative of TM is developing rapidly with new detectors and emerging technology, altogether aiming for registration of large area at high resolution. This paper, published in Journal of Microscopy, for example, introduced the multi-beam SEM, aiming to produce 60+ frames simultaneously at resolution now close to 4 nanometer. This altogether dramatically increases the acquisition time and the cost of the experiment. Our group began to concentrate effort along this line several years ago, as this paper illustrates our first attempt to use a tool originally designed for nanofabrication to produce large area registration. Fundamentally, the instruments that are designed for nanofabrication are kind of better suited to serve imaging needs because of the precision of their scanning stages and sample stability. In our case, we utilize FEI Thermo Fisher Helios Nanolab 660, which in principle is a dual beam FIP SEM scanning electron microscope. We also routinely use a retractable detector that is shown on the lower left corner, just under the pole-piece, called concentric backscattering detector to capture the backscattered electron from the sample. This slide here represents almost entire cross-section of a mouse brainstem at the level of the hypoglossal nucleus. This section is around 120 nanometer thick, and it's placed on silicon substrate. Using Thermo Fisher MAP software, we first generate low magnification image, which is usually about 600 times magnification, and this image dominates the picture here. This image provides some cellular structure, which is sufficient for visual feedback to precisely identify areas of interest. And it serves also as positioning map. Further layers are taken at higher magnification at pixel size of about 1.6 nanometers. High-resolution layers are shown in light blue squares here, as the central layer is from the hypoglossal nucleus, and the layer to the left are from the related regions of the spinal trigeminal nucleus. Thus, we capture the valuable real estate for this study at high resolution and use the rest of the navigation, the rest as a navigation tool and global positioning map. So this movie here represents the area of the hypoglossal nucleus from the adult wild-type mouse. Now, from the high altitude, you can see the boxes that represent the single field of view composing the final registration. Each image is about 3,000 horizontal pixels, and the pixel measured about 1.6 nanometers. While we are going to high resolution, we can figure out well-defined features such as Golgi and the plasmatic reticulum and synapses characteristic of the normal neuronal morphology. The resolution of this data set allows us to use well-defined cell organelles as criteria for identification of the cellular alterations in large deletion. The data set has been already partially segmented in a Rivis Vision 4D, in particular the cell bodies and the mitochondria. Please, you can point here the density. Now we are flying over the Nissl substance. This is rough endoplasmatic reticulum. Look at the well-organized rough endoplasmatic reticulum, mitochondria with characteristic cristae. On the surface of the cell, we can see different synapses making contacts with the cell body. Pretty normal Golgi apparatus. The cell, very characteristic Nissl substance with a lot of ribosomes and rough endoplasmatic reticulum. So now, I'm going to move to the similar data set that has been taken from the large deletion counterpart of the hypoglossal nucleus. And the first thing, we are going to zoom on one cell here, and we can go to the low altitude and to see the cellular morphology. Here at the intermediate magnification, one can see that the Nissl substance is dramatically reduced. When we zoom further, we can see that the Golgi is altered, and this is very typical Golgi stress. The mitochondria have less cristae, and the matrix density, the matrix electron density is at much lower level. But while we continue to fly over this neuron, we can see other changes. The nuclear envelope is still round and normal. The surface is relatively normal, but the amount of ribosomes and the rough endoplasmatic reticulum is dramatically reduced. Here we are going at the center of the field with an abnormal Golgi. And you see the mitochondria and the synapses. There are still synapses on the surface of the cell body. Now we are going to move to another neuron, but at the high altitude, you can see pretty much the frequency of these events. So now this is a neuron that, at an intermediate magnification, now you can see the irregularity of the nuclear envelope. The cell is shrunken. This is initial stages of pycnosis. The nucleus is undergoing pycnosis, but the most important now is Golgi. The rough endoplasmatic reticulum is less. The cytoplasm is a little compressed. If you see on the right side of the surface of the cell, the synapses are diminished, but they are large white areas, which represents hypertrophic astrocytes most likely. There are still synapses with different types of vesicles on the cell body, but this is typical initial stages of dark degeneration of the motor neuron. So a little further. So the conclusion here is this is Golgi that is kind of undergoing heavy stress right now. The conclusion here is, for this type of diagnostics, we need, cellular resolution, subcellular resolution at the level of a single organelle, the smallest organelle. At the same time, we need the context to find these incidences, rare incidences of these cells that are occurring. Now I'm going to move to another cell that is in a more advanced stage, and most likely this is a irreversible stage of neuronal degeneration. But again, you can see that there are, in the dark hypoglossal nucleus, there are two cases of that. This is a doll mouse. So the nucleus is truly pycnotic now. So most likely this is final stages of neuronal degeneration. The surface, few synapses left on the surface. The mitochondria heavily altered. The Golgi is under stress. Typical. All the characteristics here that we are experiencing, that we are observing right now. On the left side, another Golgi that is heavily stressed. But typical characteristic and the combination of morphological changes in the mitochondria and other cell organelles like rough endoplasmic reticulum and Golgi indicate us that most likely these neurons are dealing with oxidative stress. So this slide here presents some very interesting biometric outcomes deriving from the previous and other similar image sets. On the upper right, you can see the sample size with the quantification of the mitochondria from the single section. There is always an argument whether we cut along the axis or across the axis, so there is always a problem. But with this kind of data sample size, the things are getting kind of more definitive. So the upper row of the data represents mitochondrial density in the hypoglossal motor neurons. So the lower row of data represents the average area of the mitochondria, of individual mitochondria. The box, as I mentioned, on the upper corner represents the substantial sample size used for this study. This altogether would be challenging to collect under conventional approaches like transmission electron microscopy. The four groups that we pull together here represents P7, postnatal then 7 wild type and large deletion, and all animals wild type and large deletion. As expected with the age, the mitochondrial density in the motor neurons increased, but the area of the mitochondria doesn't change. The mitochondria at P7 large del increase in density but decrease in area. This metric indicates a larger number of smaller mitochondria and indicates clearly compromised mitogenesis. However, in adults, we observed a highly significant decrease in the mitochondrial density that is complemented by increase in the surface area of individual mitochondria. So the later increase is very consistent with the vacuolization of the mitochondria and the later stage of the postnatal development, which, taken together with the Golgi and ER stress, supports the notion of oxidative stress-related mitochondrial damage. So two-dimensional electron microscopy laid the foundation for us to identify a reliable subcellular domain primarily affected by the genetic lesion and decreased gene dosage in the large deletion. In order to fully understand the extent of cellular alterations of the large deletion, we are currently developing three-dimensional analysis pipeline. This is still work in progress, but I would like to highlight the value of this approach that attempts to complement the two-dimensional data set and actually to strengthen this two-dimensional data set. We turned into the focus ion beam, which allows us to use the gallium source to mill the sample in a very predictable way. This is shown in the left upper panel. While in a sequence of milling and electron imaging, we can produce a serial of electron images representing a volume. The lower left image represents the architecture of the electron gun and the gallium ion gun, which both focus at 53 degrees. Now the stage is tilted at 53 degrees to face the gallium beam. The gallium beam is used to dig trench into the sample and to prepare a phase for the electron beam, which is shown on the top left movie here. So further, as shown here on the top right movie, 10 nanometer, the electron beam sequentially is going to capture an image. This is a very low resolution image that is used actually to find the fiducial point and to realign the image. So essentially, this is a nanofabrication multi-step approach which requires identification of the area of interest as shown on the upper left panel, the left image, deposition of protective platinum layer shown on the upper right image, preparing a trench with fiducial points as shown on the lower left image. The final product of the preparation of the sample is a trench with a wall facing the electron beam for imaging as shown on the lower right image. So the final product of this approach is a three-dimensional volume. In this case, the volume is from the hypoglossal nucleus of a large deletion mouse. This volume represents about 630 sections taken about 10 nanometer apart from each other. And the movie illustrates the ability to dissect the volume in different dimensions. The important thing here is that this volume can be segmented. At this point, we use manual segmentation, which is pretty tedious. But after manual segmentation, we can extract different elements of this volume, and we can create these subvolumes that further can be isolated and micro-dissected, and to look for different changes and morphometric analysis. So, at the final part of my talk, I would like to highlight the results of our collaboration with the Revis and the effort to add functional context into the electron microscopy data sets. We focus on quantification of synaptic GABA transmitter because electrophysiological data indicated that GABA excitatory postsynaptic current frequency decreases in the large deletion. For GABA quantification, we devised an approach that combines post-embedding immunogol. Actually, we refined this approach that has been very popular in the transmission electron microscopy, and we adapted this approach for scanning electron microscopy by using labeling of a secondary antibody that is conjugated to 10 nanometer gold particles. GABA concentration and terminals are stochastic. The distribution is stochastic when we use very small sample size, and this is my personal experience, my personal painful experience. So therefore, we aim to increase the sample size very dramatically. But at the same time, we wanted to have resolution supporting image processing, image analysis, and image processing of 10 nanometer gold particle objects with high reliability. So this slide represents a portion of a large data set which is acquired in a similar way as I showed you before. The graph below, the data represents the intensity along the line. So colored arrows indicate gold particles and the signal they produce relative to the uranium counterstain. The intensity analysis of the image data allowed us to predict that the threshold-based segmentation will result in reliable segmentation of the majority of the gold particles. However, a small percentage of the gold particles, as shown here with the green arrow, could not be resolved based on simple intensity threshold and would require morphological approaches for separation. Moreover, the segmentation of the synaptic terminals require manual segmentation, with, in other words, identifying which profile belongs to the synapse. So considering these small challenges, we took a multi-step approach. The first step of this approach included placing the data set in the cloud and segmenting all the terminals despite their association with GABA coding particles. This step was done manually with multiple investigators involved. The synapse and mitochondria segmentation was then imported locally, where a multi-step algorithm was developed to segment the GABA-coding Golgi particles. The algorithm included a stepwise process of the image sets with global intensity thresholds followed by open filter and seed-based watershed filters. Golgi particles from the mitochondria were eliminated. Finally, the manual segmentation of the synaptic terminals and the algorithmic segmentation of the Golgi particles were intersected. The final result is a synaptic outline which can be associated with different metrics. As in this example, Golgi particles per area was used to false color the GABA intensity in each individual profile of large area taken from the wild type. Gray synapses indicate synapses displaying below threshold GABA concentrations, whereas the intensity of the profiles ranges from blue to low GABA concentrations to red for high GABA concentrations. You can note on the left image that the images, the number of individual fields that compose the merged data set. After repeating the same exercise in sample from the large division and color coding the terminals, the structured image provided immediate feedback indicating that there are more frequently blue or low GABA concentration terminals present in these samples. Here is how our data looks like as a graph. On the left panel provides an example of distribution of terminals based on concentration of Golgi particles. We applied predictive threshold based on Otsu method to set up a cutoff point for positive versus negative terminals. In general, the data supports the notion that the GABA density significantly decreases as GABA per terminal decreases and the area of the GABA profiles decreases. Altogether, this data indicates that the GABA presynaptic availability for release is diminished, which may affect in a negative way the effectiveness of the inhibitory synaptic transmission in the large division. So now, as a conclusion, what I want to highlight is that the high-resolution, large-area microscopic data offers powerful support to accept or reject well-tailored hypotheses, mainly because increasing of the power and the precision of the sample. The data achieved through more inclusive imaging has the ability to increase confidence, but also to open new opportunities not foreseen in the original experimental design. Large area, high-resolution image sets have the potential to be expanded in terms of data structuring, which adds a new dimension to create shared data libraries and collaborations. Applying such approach requires effort, and this effort is beyond and above single lab, department, and even school. Centers are important, and programmatic approach is beneficial always. Tools and instruments capable of generating such type of data are readily available. They are expensive, but if properly configured and utilized, they worth the investment. Data management and mobility is our big challenge at this point. Now, finally, I would like to thank you. First of all, I would like to thank Leica Microsystems that have been very generous supporter of us over the past couple of years. But here, this work would never be possible without the generous support of Child Health and Human Development Institute, NIH Institute, which comes to us in two forms. District of Columbia Intellectual and Developmental Disabilities Research Center, led by Vittorio Gallo, and the Program Project Grant Pathology and Developmental Origins and Prevention of Pediatric Dysphagia, led by Antonio Amancia. The work is done in the facility that I oversee, which is George Washington University Nanofabrication and Imaging Center, and this facility is supported generously by the Office of Vice President of Research. A lot of people played a critical role. Without any one of these people, this job cannot be done. The team from the Nanofabrication and Imaging Center is Cheryl, Chris Brandner, who is our senior staff scientist here for the electron microscopy. Paula Parlanti recently joined us. She has done amazing FIP-ICM. Igal is our nanofabrication senior staff scientist. He's putting a lot of effort in preparing samples with beam lithography for correlative microscopy. Our partner Chris Zugates from Arivis played a very instrumental role in providing us with protocols and analyzing data with the gold particles, also access to the web view, which is cloud-based software for structuring of the data. From the School of Engineering and Applied Sciences, and particularly Biomedical Engineering, Emilia Cheva helps us a lot with the data analysis and predictive analysis of the GABA gold particles. Liz and Pratik are two undergrad students that worked for a couple of years with us, and they structured data and did segmentations, and a terrific job. Of course, from the Neuroscience Institute and the Department of Anatomy, Anthony Lamanchi and Sally Moody leading the program for pediatric dysphagia, Beth Kropinski, David Mendelowitz, Xin Wang did the injections and biocytin injections, and Tom Maynard is very much responsible for generation of animals. Our partners from Thermo Fisher, Ken, who's been absolutely vital here for providing us the support and training on the FIP-ICM, Tim for the design of the instrumentation portfolio, Nancy Rizzo for the high pressure freezing, Sarah for the work on the confocal imaging, photon counting, wide dynamic range imaging, and the former Jonathan Boyd, who is now in the MedImmune, who played really vital role here in putting our portfolio together. So I'd like to thank everybody for joining this webinar and spending the time and listening to my story. Thank you very much.
Thank you, Anastas, for that informative presentation. Unfortunately, we have run out of time. Any questions asked today and those that are submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through August 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again soon. Goodbye.